So I think we could go ahead and get started. As I mentioned, we are going to have, question and answers throughout. I will start today's session off by giving some updates about PanKbase, and then I'll hand it over to our featured speaker, Kyle Galton. So why don't we go ahead and get started? And thank you so much for joining us today. As I mentioned, we'll start with some PanKbase updates, see if there's any feedback, and then we'll hand it off to Kyle. So just a reminder to everyone, please feel free to follow PanKbase on all of our social media. Here are the various QR codes. So we have accounts that are on Twitter, Blue Sky, and LinkedIn. So these are the various accounts there. And if you have any questions or need help with any of this, you can contact us at help@pankbase.org. Just wanted to highlight some of our upcoming dates for the PanKbase Office Hours schedule. So today is July 9th. Our next one will be August 27th, and then we'll have October 20th for the fall. And so with that, I'm going to hand it off to Kyle, who's going to be discussing about the cell states and specifically identifying the disease-associated cell states in the pancreas. So with that, thank you everyone again, and I'll hand it off to Kyle.
Okay, thank you, Mackenzie.
Thanks, Kyle, for joining us.
All right, can you see my screen?
Yes,
Okay, great.
thank you.
Okay, thanks. So, yeah, I'm going to talk today about uh, some work that the project has been doing to identify disease-associated states in the pancreas, and then also kind of detail what I mean by like cell states and cell programs, how we think about understanding their potential relevance to disease, and then also how you might visualize and interact with these programs to develop new hypotheses and understand the role of this information in disease. So yeah, does that work? Okay, great. So I'm going to start off by talking about what PanKbase provides and the platform. And one of the key things that PanKbase has been focused on is developing and providing resources that integrate pancreatic data from diverse sources. And so this is a major goal of the project is to integrate data across different sources that provide pancreatic data and create high value resources for the community that can be used for downstream analyses. And so this is one example of a resource that the project has created, which is an integrated cell map of the pancreas and pancreatic islets. And this comprises data from over 100 donors spread across different resources, so HPAP, islets generated from IDP, Proto, a variety of sources, and processes those data uniformly, collects harmonized metadata, and provides this as a resource that is a lot of value, we think, in performing downstream analyses. This resource is also described in a preprint that was recently posted on the bottom left here from the group. There's also tools in PanKbase that are available to interact with this map. So, for example, like what cell type is the gene expressed in? What are the changes in expression of that gene in disease, and also, relationship to other metadata fields that have been collected. So, like I said, like these resources represent examples of high value products that the project is focused on that incorporate data from a variety of sources that like I said, can be used for downstream analyses. And so just some examples of what types of analyses that you could do from a resource like this, like a large integrated cell map. And these are just some examples, but there could be many others. So what are the genes that have in a cell type that have altered expression in type 1 diabetes and or type 1 diabetes related traits? What is the location and activity of gene regulatory elements in pancreatic cell types and what are the genes they regulate? what genetic variants, particularly those that are associated with type 1 diabetes risk, affect gene activity in different pancreatic cell types. And then last, the focus of today is within a cell type, what are the programs and processes that have altered activity in type 1 diabetes and may also contribute to type 1 diabetes risk. So, like I said, these are just some examples of analyses that you could do with these integrated maps and combining with other resources that the project provides or other people generate, then this may unlock further insights into the role of pancreatic cell types and driving type 1 diabetes. So like I said, we're going to focus today on our work in trying to understand programs and processes within pancreatic cell types that may drive type 1 diabetes risk. And so first, I want to lay out like what we mean by a program or a process and how it fits in the context of a cell or a cell type. And so here are a couple concepts in terms of how we might build up the components of a cell. You have the basic unit, which is a gene that's measured by genomic profiling, so gene expression level, a gene. A program, the way we would think about it, sits on top of that, which is sets of genes that have either correlated or co-regulated activity within a cell type that reflects some biological process that the cell is undergoing that requires the activity of multiple genes together. Um, Cells may be utilizing different programs. So, like beta cells may have a program for insulin secretion, they may have a program for stress response, for cytokine response. And so, a cell may be utilizing different programs to varying degrees, maybe upregulating some programs, downregulating other programs. And so, what we would consider to be a cell state is at the next level a combination of gene programs that collectively make up what a cell is doing at a moment in time, where groups of cells can be in the same state. So, for example, if you perform like clustering within a cell type, you may identify sets of cells that have common features, and those common features are likely related to different combinations of programs that the cells might be utilizing. Lastly, we want to understand not only the genes and programs and cell states of a cell type, but then also the context, meaning the conditions under which a cell is in a given state. Why is it in this state? Could be something related to physiology, could be changes that occurred in progression to disease, could be in response to stimulus that the cells are exposed to, and either some physiological change or disease progression or a stimulus response may put a cell type in a given cell state. So like I said, the focus is really at the second level, which is trying to identify and understand and contextualize these sets of genes or programs that have correlated, co-regulated activity within a cell type that reflect biological processes. And as I said at the beginning, our overall goal is to identify and understand the specific programs and processes within cell types, so the pancreas, that are key drivers of type 1 diabetes pathogenesis. And I think there's a couple reasons why we feel these are important areas to focus on. So first, disease-associated programs within a cell type may represent new and important areas for therapeutic intervention. So to block the activity of a program and transition to a disease state, to reverse the activity of a program in individuals that have disease. So it has therapeutic implications in terms of reversing or modulating programs that are driving disease within a cell. Two, these disease-associated programs can provide insights into the mechanistic basis of disease. So it may help uncover novel genes that are acting within the cell type to drive disease, may uncover new transcriptional regulators of these programs, et cetera. So I think in both cases, there's value in trying to understand these programs and processes within cell types that are disease-related, both in terms of potential therapeutic value as well as an improved mechanistic understanding of disease. Okay, so there are many ways that you could go about and think about identifying programs that are active within a cell type using gene expression data. from sources such as the resources that PanKbase makes, like these integrated cell maps. So probably um, the primary way that people traditionally think about this is to use pre-existing gene sets. So these would be, pathway definitions from KEGG or Reactome or hallmark or gene ontology terms, and these are pre-specified sets of genes that have been allocated to certain processes or pathways based on literature, the known function of genes, and these are commonly used in analyses to try to understand, okay, this process is upregulated in disease or this process is downregulated in disease. But there are many limitations and challenges of using pre-existing gene sets. First of all, there's a lot of redundancy in pathway definitions and Go term definitions that can make it challenging to understand which are actually altered in disease versus those that just reflect some other process that's altered in disease. And they may also, I think, perhaps more importantly, may not reflect the actual combinations of genes that are active in a given cell type because these predefined gene sets and pathways from these databases are usually agnostic to given cell types, right? So they're general processes that a cell could be using, but they're not necessarily specifically tailored to a given cell type in the pancreas or other tissues. Um, but like I said, that would be the traditional way. I think that people think about sets of genes and then their changes in disease. On the bottom here, I think there are emerging ways to consider deriving these programs from data directly, say using single cell maps that have some benefits, I think, in terms of overcoming these challenges of pre-existing gene sets, but then also have their own limitations and challenges. And maybe there's two primary ways that you can think about this. One is where you define these gene programs directly from the data that you're working with. And there's many methods to do this. So WCGNA, for example, like weighted gene correlation networks is something that's commonly used. Methods, which I'll talk about in a minute, using non-negative matrix factorization to derive these programs, defining gene regulatory networks, like using tools like scenic and so forth. So these are all methods that derive programs directly from the data you're working with. Challenges and limitations of this approach are that it can be very difficult to separate biological from technical factors, as well as since you're inferring these programs from data de novo, it can also be a challenge to assign biological meaning to the programs that you've inferred and understand what processes that they're actually reflecting. A second, I think, a way to think about this is to infer these gene programs, but using predefined gene sets to guide the inference of the genes and their activity within a cell type. And again, there's many methods that have been developed to use pre-specified gene sets as inputs to methods to then infer what gene programs are from that starting point. Spectra is a method that we have been using, but there's others as well. And I think, that has some advantages in terms of making it easier to understand the biological meaning of the programs that you inferred relative to the inferring them from data directly. But I'd say some limitations and challenges here that, maybe there's less ability to define new or unknown programs of a cell, and also requires robust starting gene sets that you believe would accurately reflect cell biology, which would take some effort up front to be able to define what you think a good starting point set of gene sets would be to then perform this inference. Okay. So I'm going to focus mostly today on the latter point here, which is, and most of our efforts recently have been on trying to infer gene programs starting from some predefined gene set. But we've also done some work on the former, which is to infer these programs from data directly. And the method that we've been using, the methods we've been using to define these programs, all utilize a computational technique that's called non-negative matrix factorization, or NMF for short. And very briefly, the way that NMF works is that you have a starting point gene count matrix that you would derive from like a single cell map, where you'd have all of your genes on one axis and all of your cells on another axis, and the expression level of each gene for each cell. And this technique decomposes this matrix of genes and cells into two matrices, one of which is genes and factors, and the other which is factors and cells. And so what factors represent in this context are potential programs of coactive genes that we can then utilize to try to understand whether these represent processes or programs that are active within a cell type. Um, so the first matrix here, which is decomposed into factors by genes, gives you information about the importance of each gene to each factor. So for a given factor that may represent a biological process, what are the genes that are most important for that process? On the one hand, and then on the other hand, what is the importance of each of the factors or these programs to each cell? How much is this given cell utilizing a given factor that this process learned? So by decomposing this overall gene matrix into these two components, that allows us to infer these factors, which may represent programs, and then understand both the genes that may be driving these programs as well as their activity across different cells. And that allows us to try to gain some insight into the programs that a given cell type might be utilizing. Okay. And I've already covered this, but like I said, there are many approaches for performing NMF, and I've covered this back on the previous slide. Some rely on inferring the programs from data directly with no prior knowledge. Others infer these programs starting from a predefined set of gene sets. And I'm just listing here some of the different methods that have been developed to address these types of things, and each of them has different strengths and limitations, and areas where they may be more or less appropriate, but they fall into different categories of either using curated inputs, gene sets to guide the procedure, or again inferring them purely from the data directly. And like I said, what I'm going to focus on today is work that we've been doing to use curated inputs to guide the procedure of deriving these programs, using a method down here, which is called spectra, which uses gene knowledge graphs to constrain the process of deriving these factors to utilize pre-specified knowledge to then try to define programs that we hope have some more biological meaning. Okay, now just as briefly, I do want to touch on the value of curation. I think is part of the processes of doing these analyses. So, and I use analogy from defining single cell maps. So I think curation is a critical component of producing high value resources. For example, curation is often a key aspect of annotating single cell maps. You generate single cell data and you perform clustering, and then you have clusters that need to be annotated. And this annotation often relies on expert knowledge, to either explicitly, so having like manual review of the clusters to try to assign them to cell types or to cell subtypes, or implicitly, which would be maybe using literature to describe, okay, these markers of a cell type have been reported in literature, and I can use these marker genes that others have reported to annotate what I believe this cluster represents in terms of cell type identity. And so, in terms of working with single cell data and annotating single cell maps, curation is oftentimes, like I said, a major part of defining these maps, improving them, having people review them and define, additional states within these maps, and can allow you to annotate, known cell types and subtypes, annotate potentially novel subtypes based on markers and maybe some knowledge of the biology of a tissue or a cell type. It can also be used to refine clusters based on further division of cell types or subtypes. So, yeah, I think curation and expert input into defining these resources can have a lot of value and how they're then utilized. And I think we can think about the same concept in terms of defining gene programs within cell types, and the curation of potential gene programs within cell types. So, um, in the concept here of defining different cell types that exist in a tissue, where you're utilizing prior knowledge of different cell types in the tissue or cell states in the tissue. Um, the analogous situation here is that within a cell type, we can use prior knowledge of what cell types are known to do to try to define a starting point of potential gene programs that a cell type could be utilized. And so that's what we've done, which is for each pancreatic cell type, we have defined a starting list of gene sets, which have a focused number of genes, and these are all based on published sources that capture well-defined biology, we believe, of a cell type. So I'll give a couple examples here. So in pancreatic beta cells, we have various programs that have been reported in literature that beta cells utilizing. Some relate to like the core function of beta cells, so like insulin secretion, beta cell identity. Others relate to like certain stress responses that the cell type may be undergoing, so ER stress or cytokine stress, oxidative stress, and so forth. Markers of senescence, markers of cell cycle and proliferation. So each of these sets of genes, and these are just some highlighted genes here. These aren't all the genes in each program. These are just representative genes. Um, Represent curated lists of potential gene programs that a cell type could be utilizing. And so that's the example here for beta cells. And then on the right is an example from pancreatic acinar cells. And acinar cells have different functions and therefore utilize different programs. And so acinar cells, instead of secreting insulin, they secrete digestive enzymes. And there's different sets of genes that these cell types are utilizing to perform their functions. And so, yeah, these curated gene sets are the starting point for then guiding the procedure of defining programs that are active in each cell type. And just one thing to note here is that today we've been defining these gene sets ourselves, but I think these could be potential opportunities to gain feedback from others and experts in the biology of different cell types to further improve these programs. And at this point, we wouldn't, consider what we've done thus far to be definitive, but just a starting point to guide these processes. And I think moving forward, hoping to get input from others on what would be a good set of programs to represent a cell type to then guide the procedures I was just talking about. So if you're interested in participating in this process or collaborating, please let us know. And I think we would welcome input from experts and people who have interest and knowledge about certain cell types to help try to further define these curated programs that are active in a given cell type. Okay, so then we utilize these gene sets that we defined for each cell type to then, as I said, guide the procedure of performing NMF to derive these programs in each cell type. So the inputs we have are a pancreatic islet single cell map that was generated by PanKbase, and then the curated gene sets that I just mentioned for a given cell type. We stratify this map by cell type, and so for a given cell type, we have all the cells for that cell type, and then the starting point curated gene sets for that cell type. And then very briefly, in basic, but the more details for those that are interested, the workflow is that we filter cells, scale, and then select genes, run NMF using these curated gene sets as the starting point, using the spectra method, which performs NMF using these curated gene sets as a scaffold, and then output the results, quality metrics, a series of diagnostic plots, and so forth, some of which I'll talk about in the upcoming slides. So on the bottom here, the conceptual output that we get from this procedure is for each of now these factors, which represent a potential program, we have the weights of each gene and the importance of each gene to that program. And then we have some genes that were um, some genes that were in the curated gene sets that we started with, but then it also fills in additional genes that may be highly correlated or co-expressed with the starting point gene. So using that scaffold of known curated gene sets, we can learn the importance of those genes to each of the programs that the process learns, but then also fill in additional genes that may represent new information that wasn't known before, or may represent other genes that function with the genes that were in the curated gene sets. And so there's new information about potential novel genes that may be involved in these processes that we learn as well. And this is work that we've done in the project with Jason and Patrick as well. Okay, so now I'll talk a little bit about the results of performing some of these analyses focused on beta cells. What I'm showing here is after running this procedure with these curated gene sets as a starting point, this is showing the activity of each of these programs that the process learned across pancreatic beta cells. So one of the things that we derive from this process is, are all these processes actually used in pancreatic beta cells in the data we're working with. And so some processes are often used, and so they have high weights per cell, such as insulin secretion and beta cell identity, and processes that basically every beta cell is performing to some degree. To a slightly lesser degree, maybe certain stress responses that perhaps only a subset of cells are utilizing, given the context that they're in. And then other processes that may not be utilized that often by any cells, and that could be because we don't have the right context in these maps yet, and in some cases, maybe some processes are very rare or just not used very often. And so this gives us some sense for the degree to which some of these programs are utilized by cells versus others, which may be less often used. We also perform quality evaluation of the programs that we derive from each cell type. So in beta cells, I'm showing you some of the quality metrics that we consider. So first on the left here is what are the proportion of the curated input genes that are actually retained in the factors that are learned. Ideally, the factors that we learned through this process would still retain a lot of the genes that were part of the curated input gene sets. And in most cases, that is the case, that the resulting programs still retain the input scaffold genes that we're utilizing to define them. On the right, are these factors correlated with technical, meaning either technical features of the data itself or sample-based technical features, variables related to like processing of samples and so forth. And in this case, I'm showing the number of counts from the RNA-seq experiment, which is not strongly correlated with any of these programs, but in other cases, some of these programs we learned may be strongly correlated with technical factors, and therefore, you might want to flag those or not utilize those programs further in downstream analyses if they appear to be driven by technical factors. Okay, so like I said, part of I think the value of a process like this is that we can use these starting point gene sets to then fill in potentially other genes that may be involved in similar processes. And so on the left here is showing you a version of this plot here in this cartoon, which is how important different genes are to each of these programs that we learned. And so you can see the programs are learning fairly independent sets of genes. I pulled out one program here on the right, which is the ER stress unfolded protein response program. And what I've starred in red here are genes that weren't a part of the original curated gene set that were used to define the starting point for this program. So some genes that were like XPB1 and DDID3 are very well established ER stress response genes. The other genes highlighted with the stars here weren't included in the initial gene set, but if you look up the function of these genes, many of them are also known to be involved in ER stress responses, and therefore we can add genes to this process that may not have been included in the original curated gene set. And in some cases, these genes may represent novel biology, but a gene that may be involved in a process that wasn't known before. Okay, so key goal of all this work, as I mentioned at the beginning, is to try to understand which programs show evidence for potential relevance to disease. And so again, focused on beta cells, We can take all of the programs that we've derived from beta cells, starting from these curated gene sets. And I think there's maybe two main ways that we would think about trying to understand the relevance of these programs to disease. One is that do the genes involved in the program preferentially harbor genetic risk of type 1 diabetes or whatever disease you're looking at on the left. And then on the right, do these programs have evidence for having changes in expression in individuals that in that cell type in individuals that had, in this case, type 1 diabetes? Um, or in individuals that were progressing to type 1 diabetes. So the combination of those two analyses, do these programs harbor genetic risk of the disease on the left, and on the right, do these programs change in their activity in individuals that had disease or progressing to disease? I think allows us to then triangulate programs that are maybe both harboring genetic risk and also changing, and therefore maybe programs that are preferentially involved in disease in these cell types. And so in the case of beta cells in type 1 diabetes, if you look across the programs that we've learned, the very specific program here, which is interferon response and related to class 1 MHC genes, is strongly enriched for genetic risk of type 1 diabetes, whereas the other programs here show no evidence for enrichment of type 1 diabetes risk. In that same program, the interferon response class 1 MHC program is also upregulated in its expression collectively in type 1 diabetes. So together, that may suggest that program is particularly relevant to type 1 diabetes. If you reperform this procedure with type 2 diabetes, you may find other processes or enrich for type 2 diabetes risk and change in type 2 diabetes. So depending on the disease and the cell type, this may point you to very specific programs within a cell type that may be the functions that the cell type performs that are the ones that are truly disease relevant. And again, I think that's the overall goal here is to try to pinpoint what those processes are within a cell type to give people insight into how you might target these programs, or is there some new mechanistic insight that we can learn from this? Okay, so now how can we provide this information, or what would be the value, I think, of providing this information to users and the user community? And so in terms of a workflow here, if you once we've run these procedures, if you select a cell type and you can look through the gene programs that are learned in that cell type, starting from these curated gene sets and grounded in known biology of the cell type, we can provide several pieces of information. So does the program likely represent an actual process in the cell type? Meaning, is it active? Is it not, something that is likely a technical artifact? And as I just mentioned, which of the programs are enriched for genetic risk of disease, which have altered expression in that cell type and disease, and then what are the genes that are most important to that program? And I think all of these pieces of information together can give you insights into the programs of a cell type that may be disease relevant. I think there's also ways to build on this information to help individuals develop novel hypotheses about what you might do with these programs. So, for example, what may reverse a program? Are there gene knockouts? Are there small molecules that if you introduce them to cells, might be predicted to reverse the expression of a program? What are the regulators of a program? Transcription factors or other transcriptional regulators that may represent areas to modulate to knock down the program or alter its activity. And then also for genes that these processes learn that are new, what might they do mechanistically in this program and to either drive disease or is it some novel biological function within the cell type? So I think there's a lot of potential hypotheses that can be derived from this information that can guide insights into how you might target these disease relevant processes or what you might learn from them within a cell type. I think something else to highlight as well is that the way that we're hoping to set this up is to provide this information in a way that would be AI ready. And AI ready means that you're describing provenance of where these programs are coming from, a description of what it is, what was the method used to define it, what was the data that was input to it, what are the publications and sources of where this information came from, that give not only people access to more context about what it is that they're looking at, but then also informs AI tools that can help it to learn from this information for other analyses that you might want to do. Another goal here is to provide access to the analytical pipelines and that were used to derive these programs for others to apply to their own single cell data, and to recreate these processes for other questions that they may be interested in. Okay, and so lastly, I talk about visualization of this information and ways that this can be provided that are intuitive to potential users. And so I'll talk for a couple minutes about a browser for cell programs that was developed um, at Broad by Jason and Noel, that is part of the Common Metabolic Disease Knowledge Portal. And just to give some ideas for how this information could potentially be presented to user base and how they might interact with all the information that this provides. And I'm going to use here an example in the human liver that is on the site in the knowledge portal. And so, same thing for liver that we have for PanKbase and the pancreas, that we're starting from large integrated cell maps of, in this case, cell types in the liver. And what this interface that is at this URL on the top left here allows you to do is to interrogate programs that were learned from cell types in the liver by searching for a particular gene of interest. So in this case, we'll search for HNF4 alpha, which is a transcription factor that is active in hepatocytes. And if you search for that gene, it gives you information about what cell types in the liver are expressing that gene preferentially. And so, like I said, hepatocytes preferentially express HNF4 alpha. What are the cell programs within hepatocytes that express this gene? And so HNF4 alpha is a transcription factor that's important for hepatocyte identity. So it is preferentially expressed in this identity program relative to other programs. You can click on this program to obtain information about what it represents, where it was derived from, what it means for the gene that you're searching, information about how this program was defined. And so that, as I mentioned, moving towards provenance and AI readiness of these programs that provided a consistent description of what they all mean, as well as the other genes that were included in this program. And so again, in this case, when we search for HNF4 alpha, it's most highly used in this hepatocyte identity program. And this is some information about this program, how it was collected, what you can do to understand it. Okay. And then moving towards which of these programs are involved in disease risk, and for the gene that was selected for HNF4 alpha for the hepatocyte identity program specifically, what diseases or traits might that program be relevant to? And so in this case, in the liver and hepatocytes, this identity program is strongly enriched for type 2 diabetes risk, as well as genetic associations with liver enzymes, many of which are produced by hepatocyte traits as well. So this gives some insights into the diseases and traits that this program for the selected gene may be relevant to. Okay, so I'm going to stop there. And yeah, I'm happy to take any questions. I guess one point I wanted to reiterate is I think that the need again for curation is part of this process, and defining these gene sets that are inputs to these procedures, I think is very critical. And coming up with very robust sets of genes within a cell type that reflect the actual biological processes that the cell types perform. And so, as I said, we've been doing a lot of this ourselves, but if you're interested in participating or collaborating in this process for a given cell type that you may be interested in, have some expertise in, et cetera, then that would be great. It would be very welcome to have a lot of input. So thank you very much. And yeah, I'm happy to take any questions.
Great, thank you, Kyle. So if anyone has any questions, please feel free to either raise your hand and I can take you off of mute. You can use the chat or Q&A to ask any questions. All right, we have one question from an anonymous attendee. Could you comment on the process of cell acquisition altering the cell state itself, which would make it difficult to use cell states?
Cell acquisition, altering this. Yeah, I think that's a good point. So I think yeah. So basically, the process of performing these single cell assays in terms of how the cells are collected and assayed, and also like how the cells are tissues processed before the assay may alter the state of the cell. And so in some cases, you may not be looking at the true state that the cell was in prior to assay. I think that is a challenge. One way to potentially get around that, or to at least understand it, is to see which programs are strongly correlated with variables that are collected as part of the experimental process, as well as the processing of samples. So, for example, do certain states correlate with amount of time that the cells were cultured, or the amount of time that it took for the sample to be shipped, um, things like that, and may then yeah, I could like flag those as potential, programs that may have some technical confounding to them. So I think that that's potentially one way to get at, understanding which of the programs may be driven by technical factors. I think the assay part itself and how the assay may change the state of the cell, I think is a little tougher because all these assays, have to isolate cells and feed them through a process and stuff. So there are going to be some changes to every cell as part of this, part of doing single cell assays. I would say that maybe trying to relate them to, again, as a starting point, and I think maybe one advantage of using curated sets as a starting point is that you can start from curated sets that are maybe defined from other sources that were less reliant on single cell profiles, that may better reflect the actual states that a cell could be in, from literature or other studies that are, less reliant on isolation and things like this. So at least in terms of a starting point to guide this process, I think you can maybe circumvent that somewhat by having your starting point be processes that are true reflections, as far as we can tell, of the actual states of a cell. Um, and yeah, I think then like I said at the beginning, trying to, as best as possible, understand technical variation that may be correlated with the states they learn, and there may be some states that are driven by technical factors or strongly correlated with technical factors that yeah, I might you would just be aware of. I think when you interpret them and whether they're more likely to be artifacts of the sample processing or the assay that was performed, in terms of how you might interpret them. Yeah.
Great, thank you, Kyle.
I think it's a tough problem, but yeah, yes, it is a very good question. But I think, yeah, again, I think starting from some curated sets in biological processes that are actually known to be functioning within each of these cell types as a starting point, I think is another benefit of using procedures like this because you can ground the inference of these programs in things that are they're not reliant on changes that you learn directly from the data.
So we have another question in the chat which asks, are the gene sets shown here derived from differential gene expression of each of the data sets from the published resources, or is it a mix of different methodologies?
I think it's at the moment a mix of different methodologies. So it's in some cases genes that are were reported to be like, markers of given cell types. In some cases, genes that were I don't think maybe like changes in disease because I think then that would be circular and maybe that's the point of what you're getting at, right? I guess if you're defining the gene sets based on things that are reported to change in disease and then you're asking do they change in disease, that would be biased, I think, towards that gene set. I think it's more like less about, I think, and if we go like look at those studies that have reported those genes, less about these gene sets are changing in the disease that we're studying here, type 1 diabetes, and more genes that either from some prior study looking at the function of a cell type or review that has summarized known processes in the cell type that is just defining the genes based on certain processes that have been reported before. So I don't think they're not directly derived from changes in disease. And I get maybe that what you might be getting at, there's the caveat of doing that in terms of how you might interpret then a set of genes that's changing in that same disease. I think they're defined agnostic to that, but I think even when you're defining sets of genes from literature, say, genes that are involved in an interferon response, and if you're taking sets of genes that somebody else is reporting are involved in that process, they may have picked some of those genes based on things that are changing in disease. So I guess that's actually a good point. That there may be a little bit of circularity here in terms of then relating those back to disease that we should think about. But I don't think that the way that these were defined was based on directly changing in disease. No, but I think that is still, as I'm thinking about it now, a potential confounder. Yes.
Okay, and then we have one person who has raised their hand. So Eve, feel free to ask your question live.
Nope.
I'm not certain what's happening. Eve, we can't hear you. If you could type in your question, that might be a good option as well. While we're waiting, are there any other questions? Please feel free to use the Q&A or the webinar chat or raise your hand. Eve, are you able to type in your question so we can make sure it gets answered? Okay, I see here that she's saying there's a picture that she sent to delineate type 1 cell divisions. So we can take a look at that after.
That's a
Okay.
picture. You sent, oh, where you
I,
sent
I
picture?
think it must have been sent to me.
Oh, okay. Okay.
Okay, great.
Yeah, like I said, anybody who is interested in participating, who has some expertise here, that could provide input, I think that would be welcome and I think necessary to improve this process. And I think if grounded in these curated gene sets that have expert contributions and expert review, I think I think could be a very valuable resource for people. But yeah, it would definitely benefit from input from others. And if you have thoughts or sources that you'd like to share, then please do so.
Yeah, and Eve has added, we are seeing subdivisions of type 1 cross over via the pathophysiology.
Okay, great. I mean, if Eva, if you want to reach out separately, we could also talk more offline if you'd like to.
Yeah, I can connect both of you after this session. Okay. Any other questions for Kyle? I'll give it another minute or so.
Hmm?
Okay. I think that is all the questions that we have. So thank you so much for everyone who has sent in your questions and participated today. And thank you to Kyle for presenting on, the different cell types. We're very excited to see this move forward in PanKbase. And we look forward to having more office hour sessions. Our next one is going to be in August, so keep an eye out for that. And with that, I think we'll wrap up today's session. So thank you again to Kyle for presenting today as part of our PanKbase office hours. Thank you,
Okay,
everyone.
thank you. Bye now.
Bye.